Welcome to our webinar on the FDA final rule on laboratory developed tests. My name is Jonathan Genzen. I'm the Chief Medical Officer and Senior Director of Government Affairs here at ARUP Laboratories. The goal in this webinar is really twofold. In the first half of this webinar, we're going to go through a series of slides that describe in high level the details of the FDA's final rule. In the second half of our discussion here, we're going to have a Q&A where I'll be meeting with Jonathan Carr, our Chief Compliance Officer here at ARUP Laboratories, and he'll be asking a lot of the questions that have been submitted, really reflecting on what is the impact of the final rule on clinical laboratories, and maybe what are some strategies that laboratories can use to start responding or thinking about how they're going to respond to the final rule. So with that, we're going to begin with our slide deck. All right, so let's just dive right in. The FDA made its final rule on laboratory developed tests publicly available on April 29th of this year. It was officially published in the Federal Register on May 6th. And that May 6th date is really important. It's being considered the issuance date and the day by which all of the other years in the timeline and stages that we'll describe in a little bit are based on. So keep that May 6th date in mind. Technically, the final rule is actually not effective until July 5th of this year. That's 60 days after the issuance date. I do want to say one thing about the presentation as I go through it here. I'm going to use as much of the original FDA wording as is possible and practical on these slides because I deliberately want to describe the FDA perspective. I'm also including as many references as possible on the bottom of each slide. That's to the originally released document from April 29th. That's the one that the page numbers correspond to. So what did the FDA announce in its final rule? It's actually a very, very small change, only 10 words. They've changed the definition of an in vitro diagnostic product, or an in vitro device, essentially, to include when the manufacturer is a laboratory. So they're not actually introducing a definition for laboratory developed tests. They're essentially saying any IVD or in vitro diagnostic is regulated by the FDA even when the manufacturer is a laboratory. Along with that very small 10 words, the 528-page document in its preamble describes many, along with that very brief 10 words, the preamble, or the majority of that 528-page document, describes policies that are really grouped into two categories. First of all, number one, the FDA is phasing out its general enforcement discretion approach for laboratory developed tests, so that LDTs will fall under the same enforcement approach as other in vitro diagnostics. So let's pause here really quickly to talk about that term enforcement discretion. That is a legal term used by the federal government that basically says they have the authority to regulate something, but they choose not to. You can see that written there at the bottom. The FDA is now saying in this policy, they are phasing that out. They are now going to regulate or exercise oversight over laboratory developed tests as opposed to choosing not to. Part 2, the second policy, says that the FDA is adopting targeted enforcement discretion policies for specific categories of IVDs manufactured by laboratories or laboratory developed tests. What they're saying here is even though they're phasing out their enforcement discretion policy in general, there are some targeted areas where they're going to continue it, at least in part. So let's take a step back here and think about the broad concept of what the FDA has done in the final rule. They are claiming now the power to regulate all laboratory-developed tests as in vitro diagnostic devices. And they include language in the final rule, or the document, that says the phase-out policy does not in any way alter the fact that it is illegal to offer in vitro diagnostics without complying with the applicable requirements. So they're basically saying if you don't follow what we're describing here, it is illegal. And that's one reason why litigation may be considered or may be important to think about because of what that sentence really means for the clinical laboratory community. However, the FDA has acknowledged, and this is their wording, that an appreciable proportion of in vitro diagnostics currently offered as LDTs likely help patients. That's actually quite impressive that they finally acknowledged that LDTs can help patients. and that they are important to patient care. And the loss of such tests could harm patients and the associated interests. So they acknowledge that if all of a sudden they were to regulate everything, that could cause harm to patient care. So they have described that it is in the best interest of public health to expect compliance with pre-market review and quality system requirements in a more targeted fashion. And they are going to focus on laboratory-developed tests that specifically raise concerns. They haven't really described what those are, as well as new laboratory-developed tests. And so we're going to talk about a lot of these words over the next few slides. What are the quality system requirements? What is pre-market review? How do different types of tests fall under these different enforcement discretion categories? So first of all, the FDA has said they are going to continue enforcement discretion, so not exercising oversight, over several categories of tests or locations. So 1976 type laboratory developed tests, think of these at least for now as manual tests. We'll describe these in more detail. These are not subject to the new FDA rule and requirements. Human leukocyte antigen, or HLA testing specifically for transplant, is also part of this enforcement discretion. They are not going to apply these new policies and requirements to HLA testing used for transplant. Testing that is solely used for law enforcement or forensic purposes is also excluded. It's not part of the current FDA oversight proposal. The last one that has a very broad enforcement discretion category is testing performed within Veterans Administration hospitals, as well as testing performed within the Department of Defense. That's shown in the top line of this box. That is also part of this general enforcement discretion. They are not going to apply the requirements in those settings. The second line here describes a really important category for many labs, and these are laboratory-developed tests approved by the New York State Clinical Laboratory Evaluation Program. For these types of tests, there are requirements that apply. I'll describe what those are in a little bit, but they are not expected to enforce pre-market review requirements. So if you have a test that has been reviewed and approved by New York State, you would not have to submit that ultimately to the FDA unless there are certain modifications that were not approved by New York State. We'll get into this a little bit later. The third category are laboratory-developed tests that are manufactured and performed by a laboratory integrated within a healthcare system to meet an unmet need of patients receiving care within that same healthcare facility. We're going to talk about this category quite a bit because they describe in more detail what they really mean by this category. For this group, they are not expecting laboratories to submit the assays to the FDA for pre-market review, and they would be excluded from certain quality system requirements as well. And I'll describe these in more detail. You don't have to memorize this now. This next category or grouping, you're going to see on the next slide for a few other types of tests. This fourth row describes currently marketed in vitro diagnostics offered as LDTs. That's a complex way of saying a laboratory-developed test that was marketed prior to that date of issuance of this final rule and that have not been modified or that have only been modified in certain limited ways. Again, we'll get into this in more detail. This is why that May 6th date is really important. If your LDT was currently marketed prior to May 6th, the FDA does not expect that you would submit that as pre-market review in stage 4 or 5 of the upcoming timeline that we'll talk about. And it would also be exempt from quite a few of the quality system requirements that a new test may be subject to. The same grouping here is also true in number 5, non-molecular anti-sera LDTs for rare red blood cell antigens, or RBC antigens. These reagents are really tough to get a hold of, and they recognize that if they exercised full oversight over this, they would frankly cause patient harm in transfusion medicine settings. So again, these would not need to be submitted to the FDA for review, and they would be exempt from quite a few of the quality system requirements. Because the FDA has maintained now that they've had the authority to regulate LDTs, and they have chosen not to in the past, but now they are choosing to regulate them, they describe the timeline of these new requirements as a phase-out policy instead of a phase-in policy. Now, if you are in a clinical lab setting, these are brand new requirements for you, and it certainly feels like they're being phased in. But that's the explanation for the language that the FDA is using here. I'm going to walk through these stages relatively deliberately here so that we can talk about what's in each individual stage. But you're going to see more information about these stages in a few different ways throughout the subsequent slides. So within 1 year, so by May 6, 2025, labs would be required to comply with the Stage 1 requirements. These include medical device reporting requirements, requirements related to device correction and removal from the market, as well as one of the quality system requirements related to complaint files, maintaining files of complaints about assays, because that really does correspond to certain aspects of medical device reporting. So Stage 1, labs have to comply within 1 year. Within 2 years, so by May 26th, within 2 years, so by May 6th, 2026, labs would need to comply with Stage 2 requirements for their laboratory-developed tests. These include registration and device listing requirements, labeling requirements, and I think this is a really significant one. Of all the things we're going to be talking about, labeling is something that I would urge laboratories to start reading about early to make sure that you can comply within that 2-year time frame. I think of labeling as that package insert, the document that you throw away every time you get a prescription drug. There's a lot of important information in those documents, and labs would need to compile that information according to FDA requirements. As well as investigational use requirements. This is an interesting one. If your LDTs are being used in investigational studies by other individuals, there may be additional requirements that the laboratory must now follow, and these are described within the Code of Federal Regulations. We'll go into this again in a little bit more detail. I included this comment UDI, that's a unique device identifier. There are requirements specifically related to labeling that help you trace back each individual component or reagent or lot or device. This is going to be very challenging for laboratories that are frankly not physically manufacturing kits, but assembling these things as test systems. That's why I recommend people start reading about labeling in particular early on, because I think this will be challenging. Stage 3 are requirements that would need to be met 3 years after the final rule was released. So this is May 6th, 2027. These are the quality system or QS requirements that the FDA is saying will apply to laboratory developed tests. I'm going to describe what each of those ones are on a subsequent slide. So for now, let's just remember quality system requirements. This is also a big deal. It's a bigger deal even probably than labeling because there are significant requirements related to how you do your design work related to laboratory developed tests and how you document information. And that is probably different than the way that you are documenting information currently for LDTs. So this is also a requirement that labs should start reading up on early, because if you start 2.5 years from now, you're probably not going to meet the requirements by year 3. Stage 4 and 5 are when laboratories would need to submit certain assays for pre-market review. So Stage 4 are the pre-market review requirements for high-risk IVDs offered as LDTs, or laboratory-developed tests. This would be due 3.5 years from the implementation date, so this is November 6th, 2027. And then the pre-market review submission requirements for moderate risk and low risk IVDs would be due 4 years from that May 6th date. So this would be May 6th, 2028. Stage 4 are for tests that would require pre-market authorizations, high risk. Stage 5 are for things that would require that 510K or de novo review or clearance. So let's dive into some of these details and a little bit more information over the subsequent slides. And hopefully this will start coming together and make a little bit more sense. First of all, what I tried doing was compiling all of these requirements into a high-level table that we could take a couple of steps back and sort of reinforce what we've already talked about. So in the left column here are the requirements color-coded by stage. So this light blue is Stage 1, light green is Stage 2, this orange is Stage 3, and 4 and 5 are this Stage 4 and 5 are this light red color. As we talked about, these pre-1976 or 1976 type assays, HLA testing for transplant, forensic testing, and testing performed in VA hospitals as well as for Department of Defense, none of these new requirements apply in those settings. For tests that have been approved by the New York Clinical Lab Evaluation Program, Stage 1, 2, and 3 requirements will still apply. So you'll have to follow design control, you'll have to register, you'll have to list, but the FDA will not expect you to submit those tests for subsequent review if they've already been reviewed and approved by New York State. This includes New York exemption category tests as well. That's something they talk about in the final rule. For this next 3 categories of things, so testing for unmet needs, LDTs that are currently on the market, as well as LDTs for rare red blood cell antigens, the stage 1 and 2 requirements apply. So you need to do the medical device reporting, you need to do the registration, listing, and labeling, but you are not expected to comply with the quality system requirements outlined here, with the exception of record storage requirements, particularly for ongoing manufacturing activities or modifications. Inherent to kind of what we've talked about and what I showed on an earlier slide, for new laboratory developed tests, the FDA is expecting compliance with all of these requirements. They are also expecting compliance with all of these requirements for currently marketed LDTs that are significantly modified in certain ways. And we're going to talk about what that definition means in a little bit. This was a lot of information to put on one slide. If you find errors or suggested edits, feel free and reach out to me. I've included my email address on the bottom. We're going to distribute this slide separately as a PDF along with the slide deck as well for those of you that find this helpful in thinking about your LDTs and how they fit these categories. So let's go into some of these additional details for some of these categories under that subset of partial enforcement discretion. So testing for unmet needs is the first one of these. The FDA describes these as LDTs manufactured and performed by a laboratory integrated within a health system to meet an unmet need of patients receiving care within the same healthcare facility. So let's unpack that just for a moment. These are LDTs for unmet needs in a healthcare system, but only for their own patients. And they describe this limitation here in this next box, this box below. The FDA does not consider this unmet need category to include patients that are being treated at an affiliated hospital with a different corporate ownership than the laboratory. So if you're using LDTs for unmet needs and outreach purposes, you could not use them under this new described rule in that outreach setting, only for patients within the same healthcare facility and corporate ownership. The policy is also limited to LDTs that are ordered by a healthcare practitioner on the staff or with credentials and privileges of a facility owned and operated by the same healthcare system, employing the laboratory director and performing the LDT. So unpack that one for a minute. That basically means only in your healthcare system, and it's the same system that employs the lab director. Many of us can immediately see some problems with that, right? Many lab directors may be an employee of the School of Medicine and not the healthcare system, or a reference laboratory, or another community laboratory that's offering services for many hospitals. So I think they've created a very restrictive definition of unmet needs that actually is not going to be applicable in many settings in particular. They just have described what an unmet need is. I think that's a really good question. When there is no available FDA-authorized in vitro diagnostic that meets the patient's needs, that's kind of a common sense definition. But then they describe this in a little bit more detail. What does that really mean? They describe 3 circumstances. So when there is no FDA-authorized IVD for that disease or condition, for example, because it's a rare condition, or there is an FDA-authorized IVD, but it is not indicated for use on that particular patient, or there's a unique attribute that needs to be added. Category 3 here is there is an FDA-authorized IVD, but it is not available to that particular patient. That's a pretty broad one, and that may be applicable in some settings or unique circumstances. I think there's still a lot of confusion on what this means and how you can and cannot apply this. So they provide additional examples within the document. And I'm going to walk through these relatively slowly and talk through some of them. Example number 1 are LDTs for cytogenetic analysis associated with rare diseases or conditions, certain metals testing, viral load monitoring for some transplant-associated viruses, or diagnosis of certain mosquito and tick-borne diseases, but only when there is no FDA-authorized IVD for that disease or condition. So if there's an FDA-authorized test, you can't claim this unmet need circumstance. Okay, category 2 for their unmet needs examples are LDTs that are used to accommodate alternative specimen types that are infrequently tested. So when I read this, I thought, hey, here's a caveat for body fluids. This could be really helpful. But then you read the next part of this sentence. When the specimen type required for the FDA-authorized IVD is not and cannot be made available. Right? This is not how we order body fluid testing. You don't order a body fluid test because you can't get a serum or plasma test. You order a body fluid test because you want to know if there's an infection in a certain body fluid or if there's supportive evidence for a tumor marker, for example. So this one may be difficult to apply to body fluids, unfortunately. Category 3 are LDTs for use on pediatric patients when an FDA-authorized IVD is indicated for use on adults only. So I think many of us, particularly in pediatric hospitals, are going to start looking through your package inserts to see whether it's described for adults only or if there aren't any specifications in the intended indications for use specifically related to adults versus children. Category 4 are LDTs for the same indication as an FDA-cleared or approved test that is offered only in another healthcare system that is not accessible to the patient, and the developing laboratory will not make that IVD available outside its system. So let's unpack number 4 here for a second. I think what the FDA is saying is if there's an unmet need here, send the patient to another healthcare facility, develop another doctor-patient relationship, order the test there, get it done there, or try and send the specimen to that laboratory. And if you can't do either of those two things, then it's an unmet need, and then you could offer that testing or that LDT for patients in your healthcare facility. So again, very, very restrictive, not practical, and actually, to be blunt, very expensive. Example number 5 here are LDTs for emerging pathogens for which there is no FDA-authorized IVD and for which the FDA has not yet identified an emergent situation. So this is the pre-emergency use authorization pathway. I think this is a helpful example, but again, it's restricted to only your facility and only your patients. So if you're doing, let's use the next COVID as the next example. If you're doing outreach testing for an emerging pathogen, you couldn't necessarily use the unmet need for those outreach patients. They include 2 other important factors here that I'll include at the bottom. They specifically say that potential improvements in performance or lower cost in comparison to an FDA-authorized test that meets the patient needs does not fall within this policy. So if you have an LDT that costs $10, or you could send it out and pay $1,000, the FDA is saying you need to pay $1,000 and send it out. Cost itself is not a factor in deciding unmet need. And the same is true for improvements in performance. So if your test has a 5% coefficient of variation and the FDA cleared test has a 20% coefficient of variation, that doesn't qualify as an unmet need either. Lastly, the FDA has specifically said that when an FDA authorizes an IVD that meets the need of a patient, then the LDT would no longer fall within this enforcement discretion policy. So as soon as there's an FDA authorized test for that need, then you can't use the unmet need as an example. I know we've spent a lot of time on this slide, but this is a really important one. This is a really important one to hospital settings in particular, and so we'll probably talk about this a little bit later as well. The next big category that is worthy of significant discussion are laboratory-developed tests that are currently on the market. And the FDA has said that they plan on continuing this targeted enforcement discretion for LDTs currently on the market as long as they're not modified or that are modified only in certain ways. So their approach here, and this is what they describe in that preamble to the final rule, is they are going to request labeling information as part of stage 2. They're going to look at that labeling information to identify LDTs that specifically raise concerns. They can do this because labeling information includes IVD performance information as well as a summary of a supporting validation. And so there's information they'll be able to look at in theory to say, these are the LDTs we want to look at and with more scrutiny. I want to point out this third point in particular here because it leads to an interesting discussion that we may get at later. The FDA intends to look very closely at claims of superior performance and whether those claims are adequately substantiated. So in a clinical lab, right, we always talk about continuous quality improvement. Can we make our LDTs better? Can we improve patient care? That's part of, the nature of what we do. That philosophy is a different concept than what the FDA uses to review 510K submissions and de novos as well. When you think about a predicate device, substantial equivalence to a predicate device, the FDA is looking to make sure that a test is substantially equivalent to something already on the market. A claim of superior performance actually heightens the regulatory scrutiny. So our community is going to want to be careful of the language that we use as part of these submissions. And they've said this in the rule itself. These are not my words. We've already described that the FDA intends to enforce record requirements for any manufacturing activities you do for currently on the market LDTs that occur after the date of issuance. So if you're changing your assay going forward, you need to save those records of what you've done, and the FDA expects us to do that. And we've described this bottom line already on the prior table, so I'm not going to read it here. Now, the FDA, in its discussion of currently marketed LDTs, describes in more detail what types of test modifications are sufficient and that would necessitate a pre-market review or a submission to the FDA, as well as adherence to quality system requirements. Those are things that currently marketed assays you don't have to do, but if you modify significantly, now you would have to do. And they specifically say changes in the indications for use of the in vitro diagnostic. So you say it's for one thing and now you modify it and saying it's to be used for another thing. That would require pre-market review. Altering the operating principle of the IVD, and the example that they give is changes in critical reaction components. So if you're changing key reagents, however that's defined, you would have to submit that to the FDA. If you include significantly different technologies in the in vitro diagnostic, and they provide some examples of what these different technologies are. For example, if you add artificial intelligence or machine learning, you would have to submit to the FDA. If you change a molecular assay from targeted sequencing to whole genome sequencing, you would have to submit that to the FDA. If you change an assay from immunoassay to a mass spec assay, you would submit that to the FDA. Or, and this is one that makes me particularly nervous, if you change your assay from a manual method to an automated method, they have stated that you would have to submit this for pre-market review. I believe, and this is my interpretation, this could even apply to things like automating a manual ELISA or putting a PCR assay on a liquid handler, things that are relatively routine and that are done to improve quality. This would now increase regulatory review and significantly increase cost. Lastly, if you make any change that adversely impacts the performance or safety specifications of an LDT, you would have to submit that to the FDA for pre-market review. So there is a subcategory of test modifications that specifically relates to modifying another manufacturer's test. And this is a pretty common scenario in many settings. And I'm going to read through what the FDA says here because I think it's really important. The FDA is adopting a policy under which it generally does not intend to enforce pre-market review requirements for certain changes to other manufacturers' lawfully marketed tests. What that means in other words is if you make certain minor modifications in certain categories, they would not expect you to submit those to the FDA. So let's describe this in a little more detail. When a high-complexity laboratory certified under CLIA modifies another manufacturer's 510K cleared or de novo authorized test, and they're following design controls, and the modification does not significantly affect the safety or effectiveness of the test, or does not constitute a major change or modification that we described on the last slide, then you don't have to submit those to the FDA under stage 4 or 5. But keep in mind, this concept only applies to those low and moderate risk tests. If you're modifying a high risk test, so a test that has a pre-market authorization, they expect you to submit those under stage 4 to the FDA for pre-market review. And just for the same types of changes that an IVD manufacturer might make as well, they would expect you to submit those as well. So this is one reason why you may have heard about the down classification process that the FDA is doing, where they're moving many Class 3 high-risk devices to Class 2 devices. Part of that effort is to decrease the burden here as well. I've used the term de novo a couple of times, and that's worth pointing out very briefly. A de novo test is likely a low or a moderate risk test that I think the FDA terms are that could be controlled with general and special controls. But there is no predicate device to compare it to. So in the old world, you would have to submit that to a high-risk test, even if it's not really high-risk clinically, because there's nothing else to compare it to. There's a different pathway you can submit those down, de novo, that's basically an intermediate cost test. And so you may hear that term occasionally, and I think it's important to point that out here. Another big category under this enforcement discretion and general discretion are 1976 type LDTs. What are these tests? They describe these 1976 type LDTs as tests that use manual techniques without automation and without the use of software that are performed by lab personnel with specialized expertise, and they use components legally marketed for clinical use. So tests that use RUO reagents, or research use only, do not apply to this category. And these tests are designed, manufactured, and used within a single CLIA-certified laboratory that meet the requirements for high complexity testing. So these are essentially manual old assays, for the lack of a better word. They provide some examples. I think we're going to need a lot more to better understand this. These include immunohistochemistry if there is no automated preparation or interpretation. Again, I think AP is hit pretty hard by that no automated preparation. Lateral flow tests do not count as 1976 type LDTs. They call that out specifically. They do say that various tests that use staining antibodies and general purpose reagents for cytology, hematology, and bacterial infections fit this category, as well as cystic fibrosis sweat tests, certain colorimetric newborn screening tests, karyotypic tests, and tests using fluorescence in situ hybridization, as long as there's no automation or use of software. Again, in practice, many of these assays use automation and use of software as part of the process. The FDA has said they will consider providing additional guidance on certain other types of tests that may fall within this category. I've mentioned quality system requirements a few times, so let's just take a brief moment here to talk about this concept. This is particularly interesting and difficult because the FDA is in the process of updating its quality system requirements. These will take effect in February 2026 to better align with an international standard. And you may hear this term quite a bit going forward, ISO 13485, these international quality system requirements. What the FDA has said is that when a laboratory undertakes to comply with quality system requirements, the FDA will expect compliance with the quality system requirements that are in effect at that time. So if you're doing QSRs before 2026, it's the old QSRs, and if you're using them after 2026, it's the new ones. I expect this to get very confusing, but they point this out, and I think this is important to note here as well. They also note which specific quality system requirements apply to CLIA lab LDTs. It's not all of them, it's just an important subset. So I'm going to describe these here below. In stage 3, these include design controls, how you design your assays, and these are very complex and specific in some ways. Purchasing controls, including supplier controls. Acceptance activities, including receiving, in-process, and finished device acceptance activities. Corrective and preventative actions, or CAPAs, as well as the record requirements that are actually due immediately here in stage 1. I would highly encourage people to read about design controls in particular, along with general quality system requirements. And we will clearly work on providing other educational modules and webinars going forward to describe these activities, because these are different than what clinical laboratories are used to under CLIA. There's some similarities with some of these with ISO 15189 for those of you that are accredited under that ISO standard, but that doesn't include design controls. So this will be a new framework. So along with the final rule, the FDA released 2 draft guidance documents that specifically refer to immediate public health responses. What do you do in future public health threats? And I'll show the title of this draft guidance, a particularly important one of these 2 here at the top. What the FDA has said is that they do not intend to object to the offering of immediate response tests, as defined in this guidance, when the test is manufactured and offered by certain laboratory manufacturers, it's been appropriately validated, the FDA is notified, there's appropriate transparency, the test is labeled for prescription use only, and there is no EUA declaration already. So in principle, this actually sounds like a pretty good progressive concept to help us respond to public health threats more quickly. However, it's the definition of what a certain laboratory is that really cuts down the applicability of this potential new guidance. They consider this limited, and you can see this at the bottom, to certain tests and certain laboratories, such as those that are U.S. government labs, state or local public health laboratories, or other laboratories that have agreements with the U.S. government. So unless you have an agreement with the U.S. government, you couldn't use this provision if this draft guidance is finalized. So again, it's a good concept, but the devil is kind of in the details, and it is very, very restrictive and will probably prohibit many laboratories from offering tests for emergent threats prior to the declaration of a public health emergency. So I know that was a lot of information. I hope you use these slides as an easy reference when you're thinking about the final rule. There's a lot of information in the final rule, and I think 20 slides is a lot easier to digest than 528 pages. I've received a lot of questions on who do you ask for clarification on the final rule. We've got hundreds, if not thousands, of questions. What the FDA said in their webinar is please send an email to this address, ldtfinalrule@fda.hhs.gov. I hope they provide other faster mechanisms for questions because these types of questions are going to be urgent and that directly relate to clinical testing needs of patients. And it will not be appropriate to significantly delay specific responses to these types of questions. I know there will be draft guidances as well in the future, but we will probably need much more specific information much earlier than that. I'm including my email address here on the bottom. I am happy to talk about this issue with anybody. I can't provide definitive answers, but we can provide our perspective on what we think the final rule means, and maybe some areas where laboratories may want to focus a little bit more in some areas than others. So feel free and reach out to me as well. With that, we are going to transition to our Q&A, so just give us a quick moment. We'll switch our cameras over, and we will be right back.
Okay, Dr. Genzen, welcome back. It's great to be together for another Q&A session.
Thank you.
That was an excellent presentation and summary of the final rule. It is no small feat to try to summarize in 30 minutes 160 pages of 3-column, single-space, dense regulatory text. So great job.
No, thanks.
And I'm guessing there was a lot of screen capturing going on when you showed that colorful matrix, which is just a very valuable comprehensive snapshot of the rule.
And we will make that available to everybody as a separate PDF. So if you didn't get a screenshot, we'll make it available on our website.
Before we get started with the Q&A, I just got to mention that between the time of the proposed rule and the final rule, I finished this book titled 438 Days. It's a fascinating true story written by investigative journalist and author Jonathan Franklin. Great first name.
Good name.
It's an account, true story again, account of a fisherman who was lost at sea off the coast of Mexico and drifted 6,000 miles across the Pacific Ocean. Of course, ingenuity, grit, determination, and a lot of good luck got him through it. And I imagine the lab industry, we're all in the same boat, so to say, that we're in for a strange new adventure in foreign territory together.
That's a really good parallel. This has been a very strange time. We will get through it together, but it has absolutely been a journey.
Okay, let's jump into our questions.
Sounds good.
So, Dr. Genzen, are there grandfathering provisions in the final rule?
Yeah, people keep using that term grandfathering. I want to be very specific about this, and the FDA was very specific about this as well in their webinar on May 14th. There is no grandfathering in the FDA final rule. There is some continued enforcement discretion, which is what we talked about in our slide deck, where they are not going to apply certain rules currently under their plan. But a lot of how this is structured really means that the FDA could change its mind and apply further rules in the future, depending on what they think may or may not be needed. So I want to be very clear that using the term grandfathering is somewhat misleading. There really is no specific grandfathering. And even for tests like we talked about with currently marketed tests, testing for unmet needs, and New York cleared and approved tests as well, there are still requirements that apply, and those requirements are specific. So I personally don't use the term grandfathering.
Good. Does approval or grandfathering of a test in one setting mean that there are less requirements in other settings to perform the same type of test?
That's a really great question. I'm glad somebody asked that one too. There really isn't much in the FDA final rule and in the preamble that says if another laboratory has a test that has an LDT that was submitted and approved, that would then impact somehow how you can or cannot do testing in your own facility, with the exception of that unmet need provision, which says if something is approved somewhere else, then for that same use, then no longer can you use that unmet need principle. So I think it's treating labs very separately. It's treating tests very separately. They've even made some comments in the final rule and in the preamble that they're specifically not regulating small labs versus large laboratories differently, for example. So it's interesting. I could actually see many submissions under the final rule as it's currently designed for the same test or the same modifications of, say, another manufacturer's LDT for body fluids, for example. Just because somebody else has that test approved for a body fluid doesn't mean that somehow you're not subject to those requirements or that you don't have them submitted as well. The one other layer I would add to that, though, is once a predicate device is cleared or approved, it does change somewhat the review pathway or the burden on laboratories when they submit, because there is a predicate device and you wouldn't, for example, have to go down a de novo pathway. So there are a few areas when you really get through the details that describe minor differences, but for the most part, each laboratory is on its own and responsible for its own submissions.
Okay, our next question is about a term you just used in your answer to the last question. It's a term that we keep hearing about, predicate device. Can you further explain what your understanding of that term is in terms of relevance to the final rule in LDTs?
Yeah, it's a good question. I'm glad somebody brought that one up as well. And we touched on that briefly on the slide deck, but I think it's worth a little bit more clarification here. That idea of a predicate device really is how the FDA looks at new tests. When you submit a test for clearance through the 510K process, they are comparing your test to a predicate device that's already on the market. And their threshold for determining whether it's appropriate or not is that concept of substantial equivalence. The FDA is saying, hopefully, your test is substantially equivalent to something that's already on the market, and therefore they can derive this sense of safety and effectiveness from that. It's a valuable standard. It is a practical way of reviewing tests. But if you think about it in a little bit more detail, it is focused on comparison to the prior instead of innovation going forward. And I think it's one of the reasons why we've seen so much innovation in the LDT, laboratory developed test space, and maybe less innovation in the FDA cleared and approved process, because the standards are different. The cost of introducing something brand new is much higher under this FDA process than it's been under the LDT process. And that's, we'll probably talk a little bit more about innovation as we come up as well. But I think that's an important term to understand, because as you submit things to the FDA or you think about how you submit them, we talked about this in the slides as well, think about that substantial equivalence term. Think about how you're describing your test, because claims that are too boastful could actually create additional regulatory burden and risk that perhaps a test may not be cleared or approved, even if it's analytically just fine.
Okay, so our next question is, can a lab offer an LDT if there's already an FDA-approved test, similar test, I suppose, already offered elsewhere?
That's a really good question. my understanding based on the FDA final rule is that there's nothing that would prohibit a laboratory, as long as they're a high complexity CLIA lab, from submitting an LDT for review by the FDA for its own clearance or approval. You're not prohibited from doing that if there's another test on the market. There's nothing that would stop you. What we talked about in the slides, though, is if you're using that LDT for an unmet need, and there is ultimately another IVD that is approved by the FDA for that same need, then you could no longer use that unmet need provision as long as that other test was available to the patient. So there's a little bit of nuance to that, but labs are free to submit their own LDTs through the described pathway.
Okay, and this relates to another question that we got, which is, do LDTs need to be removed from the market, or should the lab stop offering an LDT if there is already an FDA-approved test or an FDA-approved test becomes available later?
Under the provisions, and we got to this a little bit in the slides, your currently marketed LDTs can stay on the market as long as you're following the requirements and those stages that the FDA has outlined, and following that pathway. So you don't have to remove a test from the market just because another test is on the market, for example, as well. But if you modify it, and that's really where things are going to get a little bit fuzzy based on how they've defined modifications, there are additional regulatory requirements if it's a significant modification. And laboratories are going to have to decide and start thinking about whether that modification is worth it and whether you can afford a submission even if you make that modification.
Okay, so our next question was about sample stability and specimen types. Are there any requirements in the final rule relevant to those?
I've been getting so many emails and questions about this. Specimen stability is interesting, and there are some comments a little bit in the preamble to the final rule that describe that. So I would search the document for the term stability or specimen stability to find a little bit of insight there. I think they're trying to use the modification concept to describe circumstances as well that may be minor modifications and that maybe would not require the submission as long as you were following the quality system and design control requirements as well with those modifications. So I would, I think, and again, you have to ask the FDA definitively on this, I suspect there are types of modifications like specimen stability that may not necessarily require submission, depending on how it's done. We talked about body fluids as well. That's a real conundrum. That is a clinical issue that's going to have to be resolved because body fluid testing is done in a lot of laboratories. It's often done using a modification of an FDA cleared or approved assay. It's one of the simplest modifications you can do. I would recommend people watch the latter half of the FDA webinar a little bit to also listen to the comments as they talk about clinicians having the flexibility to still order tests in the way that they see clinically fit. There may be some implications in that language to test modifications or to body fluids. I'm not sure I agree with that concept, and I'm not sure I agree that thinking about that in the context of a body fluid is a good idea. One thing we've done here at ARUP is we've been very deliberate about our body fluid test builds so that you have different orderables, so that you can have different interpretive comments, and so that you can separate that body fluid result away from the same analyte if it's serum or plasma, because you never want somebody to accidentally confuse a source type. So I would be very cautious about thinking about implications of test ordering practices with body fluids in ways to maybe minimize regulatory risk, because I think those introduce clinical risk, and we want to keep that in the back of our minds as well.
Right. Great point there.
Thanks.
Next question is about instrumentation.
Yeah.
Someone asked, what are the implications of buying new instruments for LDTs?
The FDA is going to have to clarify this, to be blunt. Instrument replacement is obviously necessary in clinical lab settings. When you are fortunate, you can do that in a timed manner that is predictable and that you plan extensively very far ahead. Oftentimes, you don't have that luxury because an instrument breaks and you need to change something quickly to support acute patient care needs. It is disheartening that the final rule really doesn't address a concept or pathway for emergency validations in that concept. I worry a little bit that the only alternative that they're considering is just you send the test out until you get something else validated and approved and reviewed by the FDA, which can be a very long process. That's going to need guidance. That's going to need additional clarification. That also gets a little bit to that idea of automation that we talked about as well. I fear that common automation in a laboratory, putting an ELISA on a liquid handler, for example, is something that under the final rule may have very significant implications for cost and timeline of validation studies as well. So I think of automation as a different type of instrumentation, certainly if it's analytical, but I put that in the same bucket as well. This is going to be a very big question from a lot of laboratories, and the FDA is going to have to put out guidance very quickly because instruments are going to start needing to be replaced.
I've heard you say throughout the presentation and in some of your answers already, you've talked about unmet needs, and we have some questions about that. So for the enforcement discretion policy regarding LDTs in a lab integrated within a health system, it mentions meeting unmet needs. And so what do you see as the challenges that hospitals will face with this enforcement discretion policy?
It's a really good question. I'm glad somebody has asked that as well. I don't think the final rule reflects the current structure of healthcare system and laboratory testing very well. That many healthcare facilities have affiliated hospitals that may be under separate ownership, but you're providing essential laboratory services for patients in those hospitals, even if they're not under your ownership. So I'm very concerned about unmet need testing in that setting. I'm concerned about unmet need testing in reference laboratory settings too, right? Being at a reference laboratory, people send us specimens deliberately because there's an unmet need. Otherwise, they would do that testing locally. And reference laboratories, from my understanding of the final rule, would not qualify for the unmet need enforcement discretion as well. That's mind-boggling. It doesn't make sense to me, but that's the way that the FDA has expressed it in the final rule. I want to point out as well here, though, none of this is actually in the rule itself. It's in the FDA's description of enforcement discretion. It is in their liberty to change these things at will, which is actually disconcerting and a little bit nerve-wracking because we may spend a lot of time planning extensively how to adapt to these requirements, and they could be changed because they're actually not codified, which is relatively strange. The other thing I would say, and we'll probably talk about this too, is I'm worried about unmet need testing in small laboratories as well that perhaps have less resources or maybe operating independently from a healthcare organization or a single healthcare entity, and unmet need testing may get difficult there.
Just a minute ago, you talked about automation and, for example, moving an ELISA to a liquid handler. We have a question from someone that asked, will automating a manual test necessitate pre-market submission to FDA?
That is my interpretation of the FDA final rule going forward. If you have something that is currently on the market and it is currently automated, I believe that would fit under the currently marketed enforcement discretion targeted policy. But if you're automating something manual going forward, that's new. I think that would potentially be subject to the FDA pre-market review process and other quality system requirements. Again, along with my other roles at ARUP, I'm medical director of automation. I have seen firsthand how automation improves quality. It is generally a good thing. It decreases errors. We have one of the only facilities that I'm aware of that's achieved Six Sigma quality with an automated process in a clinical laboratory setting. Automation helps. The FDA perspective, from my interpretation, is that automation is risky and therefore requires extra review. I can't think of a world where we would want to revert to manual processes in opposition to automation just because of regulatory cost. Anatomic pathologists may want to think about this concept a little bit too, though, because I know many staining procedures, for example, depend on pre-analytic automation, and they call that out specifically as something that wouldn't qualify for a 1976 type test. Doesn't fit modern practice of pathology, but that's the way that the FDA has described it. So that's going to take additional clarification, and I suspect many questions are going to come up about this very soon.
Okay. A little bit different topic here. What about flow cytometry and multiplex tests with multiple antibodies or targets? Is each antibody a separate LDT?
I honestly have no idea. We're going to have to get clarification on that from the FDA. I think the same question could be asked of panel tests and how they're offered versus individual tests. This is a common scenario, right? That is common in clinical toxicology laboratories to have assays that have the ability to detect 50 or 100 different drugs of abuse, for example. That's something we want clinically. We don't want to create extra burden on that. So we will need clarification of multiplex assays from the FDA. Maybe this already exists in the IVD market and they just haven't communicated it to the clinical laboratory market. But I think that's a really good, really important question.
Question about small labs, academic medical centers.
Yeah.
These were hot topics during the time of the proposed rule. Any exemptions for those kinds of labs?
So the short answer is no. It is interesting that they have broadly exempted the Veterans Administration hospital setting, but not other settings. That's just something that will be interesting to talk about. But they have not created exemptions for small laboratories. I mentioned this a little bit earlier in the slides, but they've specifically called out in the final rule that they expect that it is likely that small laboratories may be disproportionately impacted. That's an unfortunate thing. And I feel for people in those settings and thinking about how you comply with this going forward, because that's honestly where a lot of innovation occurs. And it's often where a lot of really clinically essential testing that's close to a patient also occurs as well. My read of the final rule is that they're trying to say that the unmet need exemption category may fit academic medical centers. But again, academic medical centers are very complex operations. I suspect it will not work according to how it's described in many, if not most, academic medical centers. And like I've talked about earlier, laboratory directors at most academic medical centers may not even be employees of the hospital. They're often employees of the medical school, and the way that they've described it may exempt certain settings as well. I hope they clarify this. I hope they make an unmet need provision that is more applicable to actually address unmet needs. I think my fear is that the current rule and how it's described in the preamble may actually have a negative impact on unmet needs, and that's unfortunate. But those two settings, small laboratory settings and academic medical centers, are not exempted.
Just a few minutes ago, you mentioned AP. Yeah. Anything else you want to mention about the potential impact to anatomic pathology?
Really important topic. For those of you in anatomic pathology who are watching, please read the final rule. Please read it in the lens of anatomic pathology, because we're going to need your expertise in thinking about how these requirements may or may not apply in your settings. Please think about whether your antibodies are ASRs, analyte-specific reagents, or if they're RUOs, research use only reagents, because that may have some implications on what exemption categories potentially could apply. I think it's been important all along to have the anatomic pathology community very engaged in this process, but we see in the final rule that that's absolutely necessary. I don't want anybody to leave this discussion thinking that anatomic pathology is less impacted by this final rule. There really isn't anything in the final rule that treats AP differently than CP. They are both subject to oversight by the FDA, with the exception of that final clinical interpretation of a slide, as long as it's not automated or there's no computers or software assisting in that interpretation. So I think that's a really important take-home point, and I hope the AP community remains and continues to be very engaged in this.
Okay, here's one that was a big topic of discussion once the proposed rule came out in October. Lawsuits, challenges against this rule from FDA. Is there anything you want to say about legal challenges to the rule?
For the audience, please note that a lawyer just asked me a question about litigation. But I want to keep this webinar current, and I suspect many changes over the next few weeks or months, in particular related to potential litigation efforts. So what I think we should do, instead of talking about litigation now, is make sure that on our website, it's the aruplab.com/FDA-LDT. We'll include this website at the end. We will keep an update there on what current litigation efforts are, if or when they're publicly available. so that people have a resource to turn to. The reason that's particularly important is this idea of a preliminary injunction. And again, I'm amazed I'm saying this to a lawyer, but there is the possibility of future litigation in which a judge could grant a preliminary injunction. That term basically means the rules would be held until the outcome of a court case. There's no guarantee that that could occur, but it is certainly a possibility. But let's not get into the details too much now. I do think that the FDA's terminology related to illegal activities has sort of forced the hand of the clinical laboratory community. It's hard to imagine continuing to operate or having scenarios where taking care of a patient in an acute need is considered an illegal activity if you can't comply with, say, the timeline or stages of what the FDA has laid out. So I think litigation is an important concept, and it's something that we'll keep people updated on.
Well said. No objections.
Sustained.
Okay, I've got 3 questions here, our final questions that are specific to ARUP.
Okay.
And first question is, does ARUP have New York approved tests on its menu already?
So the answer is yes. The vast majority of our laboratory developed tests have gone through the New York State Clinical Laboratory Evaluation Program or fit under exemption categories. So I feel like our test menu is safe currently under the final rule. Like I've alluded to earlier, I'm worried about the long-term implication of the final rule on the clinical laboratory community, particularly in hospital and small laboratory settings, which is why I keep coming back to those areas, because we want to support testing in those areas as well.
Yeah. So second question specific to ARUP is, do we here at ARUP anticipate many changes in the near future to our test menu?
The short answer is no. We're keeping our test menu intact because of our New York State approval and because of the validation work that we put into those assays and our research and development expertise. I don't see an impact to the test menu.
Okay. And really final question we have for this session is, will ARUP continue to add more tests to its menu?
We are committed to supporting our customers with a diverse testing menu that continues to innovate. I'm worried that innovation gets more expensive, in particular in all settings, particularly settings that may not have resources to support that innovation, but we are here to support our customers as well. And if I can go on a little bit of a tangent, there's something I do want to say, and this may sound unexpected coming from me, but I want to say this in the webinar as well. I was at an FDA workshop several weeks ago in Maryland, where I had the opportunity to see presentations from many FDA speakers on FDA requirements. And the speakers are fantastic. The people are fantastic. The people working there are really incredible, educated people that are dedicated to taking care of the public health through their review of in vitro diagnostics. The reason why I say that is there's going to be a lot of frustration by clinical laboratories, and it is legitimate frustration. We understand the impact of this rule on our patients, potentially, certainly if the rule stands or withstands litigation. But I want to urge people to please stay kind, not just to each other, but also to the folks at the FDA that you work with or that you reach out to, because those people are doing their jobs and trying to administer the rule. And because of the way that the FDA has approached this, that rule has the full force and effect of law. We have to comply with that, and the people at the FDA have to comply with this rule as well. So let us please work together as we navigate our way through this with the kindness that we always show each other and that we'll continue to show all of the agencies that we work with. I just wanted to say that because I think that's important. I want this to stay productive and in the spirit of a positive change, which is going to be difficult to achieve. We don't have an easy path ahead of us, but I do think we can accomplish it if we continue to stay together as a community as well.
Well, John, thank you. This was a pleasure for me to be here again with you in this Q&A session. Thanks for all you're doing for us and for the community. And thank you.
No, thank you as well. Yeah, thank you. And thank you for joining.